Greetings! Welcome to my channel, Human Evidently, where I provide evidence-based videos on human origins, paleoanthropology, archaeology, and evolutionary science for adults. They say the best way to learn is to teach, and as an anthropology student, I have found this to be true. My presentation videos are developed from the study material I build from my undergraduate courses and scholarly sources, and can be used to supplement your own learning in high school or college-level classes. This video is part of my ADHD-Friendly study guide series. Today we will be going over a study guide for Week 2 of ASM 443, Fossil Hominins. Fossil Hominins is an online lab and lecture course at Arizona State University. I attended this course in spring of 2026. Today we are covering the readings from Week 2. This video is made based on the presupposition that you, the viewer, have already read the course materials and are wanting to review the material before taking your test. If you try to use it without having read the material first, you will not get much mileage out of this presentation. Before we dive straight into this week's readings, let's do a quick recap of what you need to know this week. Week 2 focused on how paleoanthropologists interpret the human fossil record by combining anatomy, geology, taphonomy, dating, and paleoecology. First, osteology. We covered the major bones and landmarks of the skull and skeleton, including structures such as the frontal, parietal, temporal, occipital, maxilla, mandible, and key features like the supraorbital torus, zygomatic arch, and foramen magnum. Skeletal morphology is central to identifying hominins and comparing species. We also covered taphonomy, the study of what happens to remains after death. We examined how decay, scavenging, transport, weathering, burial, and fossilization shape what enters the fossil record, and why fossil assemblages are biased samples of past life. We learned the difference between relative dating, radiometric dating, biostratigraphy, and magnetostratigraphy, along with the structure of the geologic timescale, especially the Cenozoic periods and epochs relevant to hominin evolution. Finally, we covered paleoenvironmental reconstruction, how sedimentology, faunal remains, plant evidence, trace fossils, and isotopic data are used together to infer the habitats and climates in which hominins lived. If all of that made sense to you, congrats! You probably don't need to watch this video. But maybe you want to anyway, just to brush up before the reading quiz. Regardless of why you're here, I'm glad you're joining me. Let's get to recapping. So how do paleoanthropologists interpret pieces of bone and rock in the context of evolution? Most of what we know about hominins comes from fossilized remains, especially skulls and teeth. A fossil is most informative in context. Where was it found? How old is it? What happened to it after death? What sort of environment was it living in before it died? The answer to these questions lies at the intersection of osteology, geology, taphonomy, dating, and paleoecology. We need to know how to describe fossils accurately using anatomical terms. We need to know how fossils are preserved and what dating methods yield the most accurate results. We need to know how to reconstruct the settings in which our fossils lived. By becoming well acquainted with these foundations, we can gain a better understanding of why paleoanthropologists are always cautious about classification and ecological storytelling. So when we find a fossil, we're asking ourselves 5 questions. What part of the body are we looking at? What kind of organism does it represent? How did the remains get into the ground and how were they preserved? When did this organism live? And what world did it live in? All of these questions inform each other. A skull with a more anteriorly placed foramen magnum might suggest some degree of bipedal posture. But if the fossil is distorted, incomplete, or heavily reconstructed, the anatomical interpretation becomes less legit. If a faunal assemblage suggests open habitat, but taphonomy suggests that scavengers selectively removed some size classes or transported bones from different settings, then the ecological reconstruction may hold more or less bearing on interpretation. In the field, you're stacking independent lines of evidence to develop a plausible context for the fossil. One of the themes Kay Reed emphasizes is that multiproxy approaches are strongest because no single method is complete on its own. Before a fossil ever appears in a museum drawer or textbook illustration, there is a long chain of work behind it. Paleontological research begins with survey. Teams walk landscapes, map exposures, and identify promising outcrops. Once findings are made, they are cataloged, collected carefully, and recorded with detailed contextual information. As students, we're experiencing paleoanthropology as a series of finished outputs. This taxon was bipedal, this site was a woodland, this fossil is 2 million years old. In practice, the scientist makes a chain of inferences to come to those conclusions. Understanding this workflow allows us to evaluate evidence more critically and see why some claims are solid while others remain controversial. Osteology is exciting for a lot of reasons. In class, we were provided with this PDF that shows the landmarks of the cranium and mandible. The cranium houses the brain and includes major cranial and facial bones, and the mandible is the lower jaw. Many fossils are fragmentary and are described in terms of the regions preserved. You need to know how to identify the frontal bone, the parietals, the temporals, the occipital, the zygomatics, the maxillae, and the mandible. Some landmarks are more informative than others. Think about muscle attachment, posture, chewing mechanics, and the orientation of the head on the spine. Now we go into more depth on the landmarks within those regions. On the frontal bone, you'll need to know the supraorbital torus, also known as the brow ridge, and the frontal squama. On the parietals, know the parietal eminence and the temporal lines. On the temporal bone, know the mastoid process and the zygomatic arch. On the occipital, know the foramen magnum, the occipital plane, the nuchal plane, the occipital protuberance, and the nuchal lines. Ask what each feature might tell you. Is it associated with muscle attachment, cranial shape, locomotor posture, or taxonomy? The zygomatic arch relates to chewing musculature. The nuchal lines and occipital region relate to neck muscle attachment. The foramen magnum is especially important because its position and angulation are often discussed in relation to head posture and bipedalism. In broad terms, a more anteriorly placed foramen magnum suggests that the skull balances more directly over the vertebral column, which is relevant when evaluating early hominin material. The terms associated with the mandible are the mandibular symphysis, the mental protuberance, the chin, the ramus, the coronoid process, and the masseteric tuberosity. The mandible is frequently preserved and often taxonomically informative. The ramus and coronoid process relate to jaw mechanics. The symphysis and chin region are useful in comparative anatomy. Teeth are among the most durable elements in the skeleton. Teeth are great. They resist destruction better than most bones, which means they are common in the fossil record. Dental evidence can lead to understanding the age, health, diet, taxonomy, and phylogeny of the organism it belongs to. The human dental formula 2-1-2-3 tells you where the incisors, canines, premolars, and molars sit in the mouth of humans. On teeth, you need to know how to identify the occlusal surface, the chewing surface, and the cusps on a tooth. So what makes a hominin? There is a whole suite of traits that indicate whether or not a specimen is a hominin. The major traits often covered in this course are evidence of bipedal locomotion and changes in the dentition, especially reduction of the canine premolar honing complex. Some taxa also show changes in enamel thickness, although enamel alone is not a reliable diagnostic feature because thick enamel evolved in multiple ape lineages. In fossils such as Sahelanthropus and Ardipithecus, the position and angulation of the foramen magnum have been used as part of the argument for hominin affinity. But we should be cautious. Some of these fossils are fragmentary, distorted, and sometimes known partly through virtual reconstruction. The 2010 review on early hominins repeatedly shows how debates persist when evidence is incomplete or when different anatomical regions tell slightly different stories. A useful takeaway is that early hominins are often mosaics. Ardipithecus, for example, combines features associated with climbing and arboreal behavior with features suggesting some capacity for bipedalism. I am personally of the mind that technically all fossils are transitional. There was no point in time where we were ape-like and then suddenly humans. Taphonomy is the study of what happens to remains from death through decomposition, transport, burial, and eventual fossilization. It is one of the most important concepts in this module because it explains why the fossil record is never a perfect copy of the living world. Between death and discovery, a body may be scavenged, weathered, broken, gnawed, moved by water, etched by roots, or disturbed after burial. Every one of those steps can alter what survives. Most organisms do not become fossils. They are dismantled by scavengers and microbes, chemically decomposed, and dispersed long before burial. Fossilization becomes more likely when remains escape complete destruction and are buried relatively quickly in favorable settings, such as lake sediments, volcanic ash, bogs, or other contexts where destructive processes are reduced. In the technical literature, Sue and Croft stress that taphonomy is not just about damage description. It is also about evaluating whether a fossil assemblage preserves a usable ecological signal. Weathering patterns, breakage, root etching, tooth marks, insect damage, and the degree of articulation can all reveal how long bones remained exposed and what kinds of agents affected them. In other words, taphonomy is the quality control system of paleoecology. Before we infer habitat from a fossil community, we need to ask how that community was filtered. Fossil assemblages are incomplete, and that incompleteness belies a pattern. I mean to say that some animals are more likely to be preserved than others. Some body sizes are underrepresented. Some skeletal elements survive better than others. Some materials are mixed from different time intervals. Some sites include materials transported from multiple habitats. Paleoecological reconstruction may not even be worthwhile until taphonomic analyses establish what fauna might be missing and what modern analogs are appropriate. Sue and Croft add that body size distributions and ecological structure can themselves reveal bias. For example, if a fossil community is missing the kinds of small primary consumers that would be expected in any comparable modern habitat, we need to figure out if those guys are actually absent or if they're missing due to taphonomic conditions. Broadly, we cover three dating approaches: relative dating, numerical or radiometric dating, and magnetostratigraphy. Relative dating gives you the age of the fossil in relation to everything around it. It does not give you an actual age for the fossil. Numerical dating is what gives you an actual age estimate. Magnetostratigraphy matches preserved magnetic signatures to the known sequence of geomagnetic reversals. For relative dating, in an undisturbed sedimentary sequence, lower layers are older than upper layers. If a fault or intrusion cuts through a set of rocks, the feature doing the cutting is younger than the rocks it cuts. Fossils can also help correlate layers across sites when the species involved have restricted time ranges. Hominins do not make good index fossils. Numerical dating relies on the decay of radioactive isotopes at known rates. If a mineral forms or cools and traps parent and daughter isotopes in a measurable system, we can estimate the time elapsed since that event. In paleoanthropology, fossils are often dated indirectly through volcanic tuffs or associated strata, rather than by dating the fossil tissue itself. Magnetostratigraphy, then, compares the magnetic polarity recorded in rocks with the global reversal timescale. Together, these methods let paleoanthropologists build robust age models instead of relying on a single line of evidence. In the field, stratigraphy is physical and often messy. Beds can pinch out, erode, tilt, or be reworked. Radiometric dating is one of the reasons African hominin sites are so important. Many occur in regions with volcanic activity, which produces ash layers that can be dated. The Citable article explains the key idea. Radioactive parent isotopes decay into daughter isotopes at known rates, and the half-life is the time required for half the parent isotope to decay. If the relevant mineral system remained closed after formation, that decay functions like a clock. When a fossil lies between two dated volcanic tuffs, the fossil is bracketed by those ages. That is still extremely informative. It means the fossil is older than the upper tuff and younger than the lower one. In many classic hominin localities, this bracketing is what gives us precise age estimates. Stratigraphy, radiometric dates, paleomagnetism, and faunal correlation often work together. The strongest age models are those in which independent methods converge. That is exactly the same logic we saw in anatomy and paleoecology. Confidence grows when multiple lines of evidence point the same way. The geologic timescale is the master calendar that lets us place hominin evolution into deep time. For this course, focus on the hierarchy of eon, era, period, epoch, and age. Hominin evolution sits within the Phanerozoic eon and the Cenozoic era. Most of the hominin fossil record falls within the Neogene and Quaternary periods, especially the late Miocene, Pliocene, and Pleistocene, with the Holocene representing only the very recent end. Why does this matter? Because evolutionary arguments often depend on timing. Earliest candidate hominins appear in the late Miocene. Australopith diversification is mainly a Pliocene story. The later diversification of Homo is largely Pleistocene. If you do not know those broad anchors, you cannot easily situate fossils, climatic shifts, or hypotheses about the appearance of key behaviors. The International Chronostratigraphic Chart is also a reminder that numerical ages are not frozen forever. Boundaries can be refined as dating improves. So the timescale is both standardized and revisable. In that sense, it is like the rest of paleoanthropology, stable enough to organize knowledge, but always open to refinement when better evidence appears. Paleoenvironmental reconstruction asks what kind of world a fossil assemblage occupied. Reed defines three interconnected domains: depositional setting, habitat retrodiction, and climatic reconstruction. Depositional setting asks what earth processes created the sediments. Habitat retrodiction asks what sort of vegetation structure and local ecology were present. Climatic reconstruction asks what larger temperature and precipitation regime could have supported that habitat. There is no single best proxy, which is why Reed strongly favors multiproxy approaches. One category of evidence comes from geology and sedimentology. River deposits, lake beds, volcanic tuffs, paleosols, and surface traces can all indicate landscape processes and substrate conditions. Another category comes from fauna. Community composition, body size, diet, locomotor pattern, and ecomorphology can suggest forest, woodland, bushland, or grassland conditions. A third category comes from plants and chemistry. Pollen, phytoliths, macrofossils, isotopes, microwear, and mesowear all provide different windows into vegetation and feeding ecology. Different proxies capture different temporal and spatial scales. Pollen may represent a broader region, while plant macrofossils can reflect a more localized habitat. Trackways can preserve a very short-term surface event. Stable isotopes may reflect lifetime dietary patterns. Good paleoecology works by combining these scales. The field favors converging evidence over single proxy claims. Laetoli is the perfect case study for week 2 because it combines anatomy, taphonomy, geology, and paleoecology at a single famous site. It preserves Australopithecus afarensis, associated fauna, and the celebrated hominin trackways. But what makes it scientifically powerful? It's not just the trackways. Taphonomically, the Laetoli assemblage includes many isolated skeletal elements, as well as partial skeletons of small mammals. Bones show weathering, breakage, root etching, carnivore damage, rodent gnawing, and insect traces, indicating that many remains were exposed at the land surface before burial. Carnivores were clearly active in accumulation. At the same time, there is little evidence for major fluvial transport. The sediments and preservation pattern suggest that remains were scattered on successive land surfaces and then buried by volcanic ash. That means the assemblage is not perfect, but it is still considered a reasonably good reflection of a local community. Paleoecologically, the evidence from Laetoli suggests a mosaic of open woodland, bushland, and grassland elements. Open habitat taxa are important, but phytoliths, isotopes, and other lines of evidence indicate woody components as well. A hypothesis can be historically important and still need revision. The classic Savanna hypothesis proposed that the spread of open grasslands drove the origin of key hominin traits, especially bipedalism. It was attractive because it was simple and intuitive. Forests shrank, landscapes opened, and our ancestors adapted to life on the savanna. But more recent fossil discoveries and more refined paleoecological reconstructions complicated that picture. The reading on the savanna hypothesis notes that early hominins between about 7 and 4 million years ago appear to have lived in more wooded environments than the old model predicted. That does not mean habitat played no role in human evolution. It means the story was not a single-step response to open savannas. Bipedalism itself may have evolved in phases. Earlier hominins show combinations of bipedal and arboreal traits, while later, more human-like striding bipedalism becomes clearer in the context of more open environments. The modern hypothesis is that hominin evolution unfolded across heterogeneous, shifting landscapes, and that no single habitat label explains everything. Fossil hominins can only be interpreted correctly when anatomy is integrated with time, preservation, and environment. Osteology gives us the vocabulary to describe remains accurately. Taphonomy tells us how the record has been filtered. Dating methods place fossils in sequence and in years. The geologic timescale lets us see the larger temporal pattern. Paleoecology reconstructs the world those hominins inhabited. And this is all based off of a set of derived assumptions that can change as soon as new data comes out. This, my friends, is science. Thank you for spending this time with me on Human, Evidently. I hope you're leaving with a clearer understanding of the subject and a deeper curiosity about the story of life, evolution, and our place within it. Until next time, keep asking those good questions. If you want to support my channel, please consider subscribing here and on Patreon, where you can access my content for free or contribute a small donation to get your name at the end of my videos. I am working on a series of educational material for middle grade readers and 2 science fiction novels. Such endeavors are only made possible by contributions from viewers like you. Be sure to check out the sources in the description for more learning.